We've spent the last few topics talking about the process of digestion, taking in food molecules, breaking down those macromolecules into smaller bits, and then transporting that to the bloodstream. The purpose of eating, of course, is to provide energy to your body. So far, that hasn't happened. It actually takes energy to break down those macromolecules and transport them into the blood. The actual release of energy is not going to happen until this material gets to the trillions of cells that make up your body. So these molecules are going to be carried to the cells in your body where they're going to be broken down using oxygen. Basically, the molecules will be ripped apart, and the atoms that are ripped off of those organic molecules are going to be stuck to oxygen to make carbon dioxide and to make water. And that's what we're going to have a look at now. Metabolism is the sum total of chemical reactions that are occurring within your cells. Some of these reactions will require energy, some of them will liberate energy. Some of these reactions involve building up complex molecules, and some of them involve tearing down molecules. All of these reactions are linked together in a web or a network of chemical reactions. We're going to focus on the core of metabolism, which would be the reactions that liberate energy from your food. This can be broken down into three parts. Glycolysis occurs within the cytosol of your cells, and this is where sugar, glucose specifically, is broken into two molecules known as pyruvate molecules. This set of reactions does not require oxygen. The next two reactions occur within the mitochondria, and they do require oxygen to be present. The Kreb cycle or citric acid cycle is going to take place within the matrix, the center lumen of the mitochondria, and the electron transport chain involves proteins that are embedded within the inner membrane. We'll also talk about what happens if oxygen is not present. If oxygen is not present, cells can still undergo glycolysis, but they need to regenerate one of the precursors necessary for that pathway. And the process by which this is done is known as fermentation. Although the topic may seem a bit dry, I really can't stress how important these sets of reactions are. They're the fundamental basis of life. So let's take a look in broad terms at what's happening in every cell in your body. You're taking in food, of course, and that food is being broken up into smaller molecules by your digestive system, molecules that can be transported in the blood to all of your cells. You're also taking in oxygen, and the oxygen is going to be transported by the blood to every one of your cells. Inside your cells, those simple food molecules are going to be oxidized. They're going to be combined with oxygen to produce carbon dioxide and water. This is what happens when you burn a piece of wood in the fire. You are oxidizing the organic material that makes up that wood. Now, of course, your cells can't do this in one or two reactions and release a whole lot of energy instantaneously. You would spontaneously combust. Instead, what's happening is energy is being liberated from those food molecules one tiny step at a time. And the energy that's released is going to be used to take inorganic phosphate and stick it to ADP and make ATP. And then the ATP can be used as an energy source for reactions that require energy. The other thing to consider is that we're taking in food items that are complex and highly ordered. And we're ultimately breaking down those macromolecules into very simple molecules, carbon dioxide and water. So we start with a small number of large macromolecules. We break them down into a much larger number of very simple molecules. And that increases the disorder. Also, as we do this, a lot of the energy conversion is not all that efficient, and a lot of the energy is lost as heat, which also leads to disorder. Heat is simply the movement of molecules. When you measure the temperature of something with a thermometer, you're measuring how often molecules or atoms collide with the thermometer and how much energy they have, how much kinetic energy. So temperature is really a measurement of the average speed or kinetic energy of molecules. So we start off with ordered molecules. We end up with less ordered molecules, simpler molecules, and we liberate a lot of heat, which increases the disorder around us. Cellular respiration refers to the chemical reactions that liberate energy from organic molecules. This happens in every single cell in your body. And when you think about it, all of your major organ systems, with the exception of the reproductive system, are there to allow cells to undergo this process. The digestive system, of course, is going to take in food items that can be broken up and then passed into the bloodstream. The respiratory system is going to take in oxygen so that cells can oxidize the materials they got from the digestive system. The respiratory system is also going to take away carbon dioxide, which is a byproduct of this. The circulatory system connects all the systems together, and it's going to transport nutrition and gases in the blood to every cell in the body. The endocrine system does a lot of different things, but one of the major things it does is maintain homeostasis. So it makes conditions preferable for cellular respiration and other metabolic activities in the cells of your body. The urinary system is going to remove toxins that are a byproduct of the breakdown of materials during metabolism. The nervous system and the muscular system, well, they're going to make it possible for you to breathe and to eat and to pump blood, and also they're going to make it possible for you to hunt or gather food, or at least make a trip to the supermarket. A metabolic pathway is a series of chemical reactions that are linked together. We start with a precursor, and then that, through a series of steps, is going to be converted into a product. That's our end result. In this example here, we've got a very short metabolic pathway. Most metabolic pathways contain dozens of steps, but we start with this precursor molecule, whatever it is, we're calling it A, and we have a chemical reaction that converts it to a new molecule. So we have the precursor at the beginning, the product at the end. All the intermediate molecules in between are known as intermediates. So now we have intermediate B. Now that reaction had to change the molecule in some way. Maybe something was cut off, maybe something was added like a phosphate, or maybe the atoms were rearranged. That reaction would have been catalyzed by an enzyme. Remember that it's very uncommon for any reactions to occur in a cell without the help of an enzyme. And typically, each one of these steps is going to be performed by a different enzyme. Enzymes usually catalyze just one certain type of reaction, or sometimes a handful of very similar reactions. That product might be used to build something, or that product might be a precursor for another metabolic pathway. For that matter, any of these intermediates could be a precursor for another metabolic pathway. Metabolic pathways are linked together. And we can see that here. This diagram represents several different metabolic pathways, and they're all linked together. Every dot that you see represents a molecule, and a line connecting two dots represents a chemical reaction, so a change. A molecule that we're interested in today is glucose, and glucose is shown by this dot right here. This center pathway that's labeled carbohydrate metabolism is the first set of reactions that are going to liberate energy from glucose. So that straight line down towards the bottom is glycolysis. And at the bottom here we have this loop, so this series of reactions that occur in a cycle, that's the Kreb cycle or the citric acid cycle. But I want to draw your attention to the fact that glucose is a precursor for carbohydrate metabolism, but notice that there are other lines that lead into glucose and lead out of glucose. Glucose can be manufactured. Glucose can be the end product of some metabolic pathways, a pathway known as gluconeogenesis. Don't worry about that, but we talked about before the fact that if glucose is not present, if you don't have any carbs, your body can make glucose, and it can make glucose from glycerol. But also note that this pathway has branches coming off. So let's say a particular cell in your body has enough ATP for what it needs to do, but it's lacking, let's say, what it needs to make nucleotides. What it can do is it can take this intermediate here And instead of this intermediate passing down the chain of carbohydrate metabolism, it can draw this out into a series of reactions that will lead into nucleotide metabolism. So again, my point here is that all of these pathways are connected, and what determines what pathway is going to proceed is the needs of the cell. If the cell needs lipids and it doesn't need ATP all that badly, it'll pull things out of the carbohydrate metabolic pathway and build lipids. Now, on the other hand, if it doesn't have enough glucose, it may be able to pull intermediate molecules out of another pathway and shunt them in somewhere. So it might be able to take the intermediates in the amino acid metabolic pathway, pull the intermediates out and shove them in as intermediates in the carbohydrate metabolic pathway. We can represent metabolic pathways in a number of different ways. I kind of like what someone's done here. It's rather creative. They're representing these pathways as potential tube lines or subway lines in the London Underground. And what you're seeing here in their depiction, we've got these little crossovers here. That's where if you were on a subway going from station to station, you could switch to another line. So they're representing different metabolic pathways as train lines, and these transfers are where those different metabolic pathways are connected. Okay, so some quick fundamental high school chemistry. If you've forgotten this, take some time to refresh your memory, and of course ask me questions if you're still struggling. Electronegativity is a very important concept when it comes to metabolic reactions or any kind of chemical reaction. Electronegativity is the affinity that an atom has for electrons. So let's take a look at this table here, and we'll look at oxygen first. You can see that oxygen has an electronegativity of 3.5. Now, we're not going to concern ourselves with what this number means and how it's derived, but it just means that oxygen has a higher affinity for electrons than any atom that has a lower number. Let's compare that to hydrogen. Over here, you can see it has a much lower electronegativity with a value of 2.1. What that means is if oxygen and hydrogen come together to make water, for instance, oxygen is going to pull electrons away from the hydrogen. Now, in the case of water, it's not going to strip the electrons away completely. They're going to form a covalent bond. And remember, a covalent bond is where two atoms are sharing a pair of electrons. But the electrons are going to be pulled closer towards the oxygen. And as a result, the hydrogen has a slightly positive charge, not a full positive charge, but a slightly positive charge. And the oxygen in a water molecule has a slightly negative charge. So that's known as a polar covalent bond, where electrons are shared unequally. And you can figure out how electrons are going to be shared between any two atoms using these electronegativities. Chemical bonds have what's called a bond energy, and that's the amount of energy it takes to break that specific bond, and it's also the amount of energy that's given off when that bond forms. And this is really important. When we are breaking down food, we have to put in energy to break those molecules. But for the most part, we're breaking non-polar covalent bonds, and they have low bond energies. When we take those atoms that we pulled off the food and combine them with oxygen, we form polar covalent bonds that have higher bond energies. So we have to invest some energy to break the bonds initially, but the new bonds that we form give us more energy back. So we get a net gain of energy. So looking at the chart here, the rows that I've highlighted in green, those correspond to non-polar covalent bonds. So of course, if we bring carbon and carbon together, they have the same electronegativity. It's going to be a non-polar covalent bond. If we bring a carbon and a hydrogen together, they don't have the exact same electronegativity, but it's quite close. So that will give us a non-polar covalent bond. What I've shown in red here represents the polar covalent bonds. So we've talked about oxygen to hydrogen already, and you can see that it has a rather high bond energy of 111 kilocalories per mole. And at the bottom there, you can see that a carbon to oxygen double bond has a very high bond energy of 192 kilocalories per mole. Your food items contain mainly non-polar covalent bonds. So as I said, it doesn't take much energy to break those. And ultimately, you're going to produce water and carbon dioxide, which have high bond energies. And you get more energy back when those new bonds form. Let's take a look at this in action. In any chemical reaction, we've got two steps. The first step is that bonds need to be broken. The second step is the formation of new bonds. So in this example here with propane, we can see that the first step involves breaking the propane molecule apart, and the second step involves taking those bits and pieces and forming new molecules. So imagine you have a gas barbecue, you turn on the propane, and then you apply a spark. The spark will provide enough energy to break the carbon-to-carbon bonds and the carbon-to-hydrogen bonds within the propane molecule. It'll also provide enough energy to break the double bonds holding oxygen atoms together as diatomic molecules. Now that all those atoms are free, they can recombine into simpler molecules, and we get our carbon dioxide and our water. We get an increase in entropy as well. This is a reaction that liberates energy, and that energy is going to be expressed as heat and light, and that's the flame. So this is the reaction that creates flame. That reaction will continue as new propane comes into your barbecue because that heat and light provides the energy to now break apart the next set of propane and oxygen molecules. The two molecules at the bottom of this diagram, the propane and the oxygen, are our reactants. The two molecules at the top, water and carbon dioxide, are our products. And we know the bond energy for each of the types of bonds found in the reactants and products. And we can use this to figure out whether the reaction is going to liberate energy or whether it's going to require energy. At the top here, we've got the balanced equation for the reaction. It has to be balanced, of course, or the math isn't going to work. Down the bottom, we have calculated the bond energies for the reactants and for the products. So looking at the reactants, within propane, there are 8 hydrogen to carbon bonds. They each have a bond energy of 98.7 kilocalories. 8 times 98.7 gives us 790. So we do that for all the bonds we need to break. We get a total of 1,536 kilocalories. That's the amount of energy we need to put in to break those bonds. On the other side, the product side, we once again can add up all of the bond energies. So for instance, we have 6 carbon to oxygen double bonds. They have very high bond energies, 192 kilocalories. That gives us 1,152 kilocalories. Again, if we add up the bond energies for the products, we get 2,040 kilocalories. The last thing to do is to subtract the bond energies of the products from the bond energies of the reactants. And when we do that, you can see down the bottom there, we get negative 504 kilocalories. We always do the math in the same way. If the value is a negative, that means that energy was liberated by the reaction. If the result had been a positive number, that means we're dealing with a reaction where we had to invest energy. Nutrient molecules that our cells consume and oxidize have potential energy. There's energy stored within the bonds. Now, technically speaking, the bonds of your food molecules don't have a lot of energy, but the potential comes from the fact that we can pull those bonds apart fairly easily, and we can form new bonds that release an excess of energy. So for instance, the triglycerides that you have in your fat cells, they have a lot of potential energy. They're a great way to store energy because they contain nothing but non-polar covalent bonds. Those bonds can be broken, used to make water and also carbon dioxide, which releases a net gain of energy. The energy that we liberate from our food has to be transferred to the molecule we've talked about quite a bit already, ATP. So ATP will be built from inorganic phosphate and ADP. It'll trap some energy, and it only serves as a very short-term storage molecule, but it can shuttle that energy to reactions that require energy. Any chemical reaction consists of two parts: an oxidation, which is where electrons are lost from an atom, and reduction, where an atom gains electrons. So electrons are going to be removed from one atom and stuck to another atom. Now that might not always mean that the atoms are lost completely, pulled off completely, and then grabbed onto by another atom. It could be that we've got a polar covalent bond forming where electrons move closer to the atom that has the higher electronegativity. So in that case, the atom with the higher electronegativity would be reduced because it's gaining those electrons, even though it's not stripping them away completely from the atom that was oxidized. You might have heard of this saying before in chemistry: Leo says grr. So Leo the lion, of course, is going to grr. Leo, loss of electrons is oxidation. Grr, gain of electrons is reduction. Nice way to remember it. So here we have a simple example here of what's called a redox reaction, because we're linking oxidation and reduction together. We've got molecule A with an electron that's going to be transferred to B. A is oxidized because it lost an electron. B is reduced because it gained an electron. But again, it's not always the case that the electron is completely stripped away. We can still talk about a redox reaction when we end up with a bond that is polar covalent and the electrons are shared unequally. Here we have an example where iron is going to combine with oxygen and water in the air and form hydrated iron oxide, also known as rust. So when a piece of metal is rusting, this is the reaction that's occurring. It's an example of a redox reaction because electrons are being pulled away from the iron atom and they're being pulled closer to the oxygen. Here's another common example, and in this case, electrons have been removed completely. If you bring sodium and chlorine together, you get sodium chloride, which consists of two ions because the sodium has completely lost an electron and the chloride has gained that electron. So the chlorine atom is going to become reduced and the sodium atom is going to be oxidized. Let's say we're burning methane, CH4. If we burn methane, electrons are being pulled away from the carbon and hydrogen when we form carbon and water. And the electrons are being pulled closer towards the oxygen in those products. So what you're seeing here is a diagram that's just trying to show the location of the electrons that are being shared between these different atoms. In methane and in diatomic oxygen, the electrons are being shared equally between the participants. So these are non-polar covalent bonds. And then in the products, in carbon dioxide and water, we've got strongly polar covalent bonds. The electrons have been pulled towards the oxygen. And whenever we go from a situation where our reactants have non-polar covalent bonds to products that have polar covalent bonds, there's going to be a release of energy. This is what happens when we oxidize things by burning them. This is also what happens in our cells when they metabolize glucose and other food items. So we've got glucose here, C6H12O6, and it's going to be oxidized when it comes into contact with oxygen. It's going to be broken down into carbon dioxide and water. The oxygen is reduced. We say it's a reducing agent, and it's going to grab onto those electrons in the product molecules and hold them closer to itself. We also get energy liberated, and that energy is going to be used to make ATP. Transfers of electrons are ultimately responsible for the liberation of energy that's used to make ATP. Quite often there's sort of a middleman though. The electrons that are liberated in a set of reactions can be transferred to another set of reactions by an electron carrier. So in this example here, we've got an organic molecule, a food molecule, that's going to lose electrons, and the electrons are going to be transferred to an electron carrier that becomes reduced. It gains the electrons. In the process, hydrogen protons can be transferred as well. This is a reaction known as a dehydrogenation. So in this example here, the electron carrier is NAD positive, and it's actually gaining 2 electrons. One free electron that's going to neutralize the charge, get rid of that plus sign, and another electron that is coupled with a proton, which of course is a hydrogen atom. So it becomes NADH. So what we did was we transferred one complete hydrogen atom to NAD positive and then split another hydrogen atom into a proton, H positive, and the electron, and transferred that electron as well to NAD positive. You can see that process here in a bit more detail. NAD plus, nicotinamide adenine dinucleotide, is used as our carrier. And incidentally, I don't expect you to be able to write out that name, just know it as NAD plus. It's a derivative of niacin, in fact. Niacin is something you might note on your vitamin bottles. This is what it's used for, one of the things it's used for. Anyway, we've got this NAD positive, which consists of a modified nucleotide, adenine. And you can see we've got adenine, and then at the top there in white, we've got this ring structure attached to it. And that ring structure contains a nitrogen that is missing an electron. That's why it's N+. What's going to happen is 2 hydrogen atoms are going to be split into electrons and protons. One of those electrons is going to be taken in by that nitrogen to fill its valence shell, and it loses that plus charge. It becomes neutral. The other electron is going to recombine with that proton to form a hydrogen. That hydrogen is also stuck to the ring. And then we have a free hydrogen proton just floating around. So we end up with NADH, we've gained a hydrogen, we've neutralized that charge, and then we've got a free proton as well, which will also prove to be useful, as we'll see. So in a dehydrogenation, we have the transfer of an entire hydrogen atom, but remember, there's an electron in there, and then we may have transfers of other electrons as well. The net result will be that the organic molecule that's serving as the food item is going to be oxidized because it's going to lose electrons, and the carrier is going to be reduced because it gains electrons that can then be carried to another set of reactions. ATP is our ultimate energy shuttle. Remember, when you pull a phosphate off of ATP, energy is released. Once again, this reaction has 2 steps. It actually takes a little bit of energy to break that phosphate off, but the phosphate immediately combines with oxygen to form inorganic phosphate, and that process is what releases the energy. Going the other way, taking inorganic phosphate and adding it to ATP requires energy, and we store energy by doing that. Energy liberated by pulling a phosphate off of ATP can be used to do a lot of very different things in the cell. What generally happens is that phosphate is stuck to a molecule that needs to be energized. That's something known as phosphorylation, when you stick a phosphate onto another molecule. So in the example at the top there, we've got a membrane protein that acts as a pump. It can move things across the surface, but this is an example of active transport, and it requires energy. The phosphate is stuck to that pump, and then the phosphate is pulled off, and when it's combined with oxygen to make inorganic phosphate, the energy that's released can be used to pump a solute across the membrane. In the second set of diagrams, we've got a motor protein that can change shape when it receives energy, and it can move, in this case, a vesicle. It can move it along a microtubule. And this diagram is by no means to scale, and there would also be millions of motor proteins involved in this movement. Motor proteins are used in muscle contraction, which we'll talk about in great detail very soon. But anyway, you can see that the phosphate has been stuck to the motor protein. When it's released, that releases energy that can allow the motor protein to move. In a lot of reactions that require energy, what happens is an enzyme comes along and it sticks a phosphate to one or more of the reactants. And then when those reactants come together, the phosphate is pulled off. That supplies the energy needed to make this rearrangement of atoms and build this new molecule. ATP can be generated in one of 2 ways. The simplest is substrate level phosphorylation. This is where a phosphate group is pulled off of one molecule and stuck on ADP to make ATP. So you can see in this example we've got a 3-carbon molecule. The oxygens and hydrogens aren't shown, but we're taking a phosphate off of that molecule and then sticking it directly to ADP. This process doesn't require any oxygen. The second way we can generate ATP is a little more involved, and this is known as oxidative phosphorylation. This is what occurs during the electron transport chain within the mitochondria. Oxygen is going to be the final acceptor of electrons, so it has to be present. But electrons are going to be bounced along a series of proteins embedded within the inner membrane, and that energy from those electrons is going to be used to pump hydrogen protons into the space between the two membranes of the mitochondria. So we set up a proton gradient, and we're going to use that gradient to generate ATP. And we'll talk about that in a bit. There's several steps to this. So here's that substrate level phosphorylation. There are a number of enzymes in your body that transfer phosphates, and that's all they do. One group of these is known as kinases, which you might have heard of. Don't expect you to know about just yet, but we may come back to those. Anyway, we've got an enzyme here that can grab onto a substrate that has a phosphate, and it can also grab onto ADP, and it can transfer the phosphate to the ADP. In oxidative phosphorylation, again, we need oxygen. That's important. And what we're doing is we're taking electrons from electron carriers, and we're going to pass them along an electron transport chain and generate a hydrogen proton gradient. So two key things here. Electrons are going to be carried into this series of reactions by electron carriers like NADH, and we need oxygen. Oxygen is going to be the final acceptor of these electrons. We have to get rid of the electrons that we carried in to this transport chain, and we get rid of them by sticking them to oxygen and making carbon dioxide and water. So as an overview, the first step in this 3-part series of events is glycolysis, and that occurs in the cytosol, the liquid portion of the cytoplasm, and it liberates enough energy to make 2 ATP through substrate level phosphorylation. If oxygen is present, then we have 2 more steps that can occur within the mitochondria. The first is the citric acid cycle, which is going to pull electrons off of organic molecules and stick them to electron carriers. And the last step occurs within the inner membrane of the mitochondria, and it's going to use the energy from those electrons to set up a concentration gradient of hydrogen protons, and that's oxidative phosphorylation. Now, if there's no oxygen present, we still have to get rid of electrons, because even during glycolysis, electrons are stuck to electron carriers. They would normally be carried into the mitochondria, but that's not going to happen. So instead, the electrons get stuck to pyruvate, which is one of the end products of glycolysis, and that's used to make ethanol or lactate, and that's known as fermentation. In us, it's lactate that's generated. I know this topic can be overwhelming, and perhaps the last few slides I talked about went over your head. But that's okay. We're going to come back to it, and we'll go through it in a bit more detail. But what I want you to focus on is the major inputs and products of each of these 3 events: glycolysis, the Kreb cycle or citric acid cycle, and the electron transport chain. So what goes in, what comes out? How many electron carriers are made? Where do they go? That sort of thing. How much ATP comes out of each of these processes? So this diagram here is a really good one. It's a really nice summary. Take some time and learn this diagram. You can see that the vast majority of ATP is going to be generated by the electron transport chain within the mitochondria. What I'm going to do now is I'm going to break down each of these steps in a bit more detail, starting with glycolysis. Here's the greatly simplified overview of glycolysis. We start with a glucose molecule, which is a 6-carbon molecule. It's going to be broken down into 2 3-carbon molecules. and those are known as pyruvic acid molecules. In the process, we're going to generate 2 ATP. We get a net gain of 2 ATP. We also get a net gain of 2 NADH, and that's an electron carrier, remember. It's going to carry electrons to the electron transport chain. Now don't be frightened by this diagram. I'm not going to have you remember the names of these intermediates. I just want to point out that there are lots of intermediates, and most of the reactions, which are shown by arrows here, are catalyzed by different enzymes. The general process, we're going to take glucose and we're going to split it in two and then rearrange to get 2 pyruvate molecules. So the first set of reactions at the top there, notice that they require ATP. We have to invest 2 ATP molecules for every glucose molecule that we start with. But then the second set of reactions liberate 4 ATP. So we get a net gain of 2 ATP. And those first series of events are just rearranging glucose into a symmetrical molecule that can be split. Split into 2 molecules called glyceraldehyde 3-phosphate, or G3P. Again, don't worry about the name of that, but we have to invest energy to get to that state. And then afterwards, we get some ATP back. And this is the summary I want you to know. This is pretty much all the detail I want you to know for glycolysis. I want you to know that we can split it into 2 stages. The energy investment phase, where we have to invest 2 ATP to rearrange the glucose molecule so that we can split it. And then the second part, we're going to get 4 ATP out, so we're going to have a net of 2 pyruvate. And we're also going to get 2 NADH. And of course, our product is pyruvate. So what I expect you to know, you start with glucose, you invest 2 ATP, you get 4 ATP back. So you get a net gain of 2 ATP, you get 2 NADH, and you get 2 pyruvate. That's it. I'm going to go through and show you some of the steps and discuss them just so you can kind of get a sense of what's going on, but I don't expect you to know the names of the enzymes involved or the names of any of the intermediates. We start with glucose. Remember, you don't have to fuel your cells with glucose. You could fuel them with amino acids or glycerol or fatty acids or nucleotides, whatever. In that case, what's happening is that molecule is going to be rearranged into one of the intermediates somewhere in the cellular respiration pathway and just shunted in there. But we'll start with glucose, and I'm going to look at the first step in a bit of detail. Again, you don't need to know any of the enzyme names or any of the intermediate names. I just want to give you an example of the kind of reactions that are occurring. We have an enzyme called hexokinase. A kinase is an enzyme that transfers a phosphate to a molecule. It's going to take the phosphate off of ATP and transfer it to the number 6 carbon of glucose to make glucose 6-phosphate. Hexo also means 6. So the names of these intermediates and also the names of the enzymes, as scary as they might be, they actually make sense. So if you ever find that you have to learn all these names for some reason, just keep that in mind. Just break down the name and figure out what it means. Isomerase, for instance, that's an enzyme that takes a molecule and just rearranges it without changing its chemical formula. Anyway, we've got 2 ATP that are required because they're giving up phosphates. The molecules are going to be rearranged until we get a symmetrical molecule with a phosphate on each side. That's the fructose 1,6 bisphosphate. And then that can be split into 2 slightly different molecules. The only molecule that continues on to the payoff phase is glyceraldehyde 3-phosphate, G3P. The other molecule that's made is converted into G3P by this isomerase. In fact, it's just constantly grabbing onto molecules and changing them back and forth between these 2 variants. But the G3P will be grabbed by another enzyme and be pulled on to the next set of reactions. If it seems like a rather inefficient way to do things, well, it is, but that's life. Now we're seeing what happens to those G3P molecules. We have electrons being pulled off that are used to make NADH. So that's going to be an electron carrier, and it's going to carry electrons onto the electron transport chain. We also have those phosphates being pulled off and used to make ATP. Now, one thing to keep in mind is we only get one NADH and 2 ATP for each molecule of G3P. But there's 2 molecules of G3P for every 1 glucose that we started with. So that's where the numbers come from, if you're a bit confused. The numbers that are provided here are for 2 molecules of G3P. So we end up with this, and again, this is all that I want you to know for glycolysis. I just went into a bit more detail so you can get a sense of what's going on. So we have 2 pyruvate molecules resulting from every starting glucose molecule. And I should mention that glycolysis literally means sugar split. It's the process of splitting sugar. The pyruvate are going to enter into the mitochondrion because that's where the next 2 stages of cellular respiration occur. But the entire pyruvate doesn't enter. Just this part here. This is known as an acetyl group, and it's going to be stuck to a very large molecule known as coenzyme A. And coenzyme A is just going to allow this to be transferred. Coenzyme A is not going to go into the next series of reactions. Instead, coenzyme A with the acetyl group is going to be transported across a transport membrane into the matrix, the middle of the mitochondria, and then it's going to release that acetyl group. The other part of the molecule is going to be converted into carbon dioxide. So we're going to gain an electron here and lose this, and then make another bond here, and then now this here is a carbon dioxide molecule. And that accounts for some of the carbon dioxide that you breathe out. The mitochondria is usually described in almost every textbook as being the powerhouse of the cell. On a test or exam, I expect you to say a little bit more than that. After this lecture, you'll be able to describe in great detail, hopefully, what's going on in the mitochondria. But this is where the majority of the potential energy trapped in your food is liberated and used to make ATP. The mitochondria, or mitochondrion, if we're talking about just one, consists of 2 membranes. We have an outer membrane that's smooth. We have an inner membrane that is thrown into lots of folds known as cristae. And the cristae just help increase the surface area of that inner membrane. The inner membrane is mostly protein, which is kind of unusual. It's about 3/4 protein, and the rest is phospholipids. Those proteins are proteins of the electron transport chain and also ATP synthase, which we'll talk about in a moment. The citric acid cycle occurs within the matrix. That's the innermost lumen of the mitochondria. Again, oxidative phosphorylation via the electron transport chain occurs within the inner membrane. The citric acid cycle doesn't generate very much ATP. Instead, it generates quite a few electron carriers that are going to be transferred to the electron transport chain. The electron transport chain will generate 90% of the ATP we get out of each sugar molecule. Again, I don't want you to know the names of the intermediates or the enzymes involved, but I do want you to know what's coming in to the citric acid cycle and what's leaving, what's being produced. So we have our acetyl group bound to coenzyme A. Coenzyme A is going to release it, and it's going to become attached to this molecule known as oxaloacetate to form something called citrate. And that's where the name comes from for this cycle. In this diagram, they're showing in red or pink the carbons that are coming in. And you can see what's going to happen is that citrate is going to be rearranged into isocitrate, etc., etc., etc. We have rearrangements of this molecule back to oxaloacetate. In the process, 2 carbons are lost, and you can see they're being pulled off as carbon dioxide. Now the next cycle, they're showing those carbons that came in originally in blue. So the carbons that come in have to go around one full loop, and then they get pulled off as carbon dioxide the second time around. Now, also in the process, electrons are being pulled off of these molecules and stuck to NAD positive to form NADH, and that's going to carry electrons to the electron transport chain. Also, you can see in step 6 here, we have another electron carrier being made, and that's FADH2. So when we manufacture NADH, remember we're sticking one complete hydrogen, so an electron and a proton, and then another electron to that NAD positive to make NADH. With FADH2, we're just sticking 2 complete hydrogen atoms to FAD. So we're carrying 2 electrons, but also 2 protons. So the whole point of this cycle here is to pull electrons off of these food molecules so they can be passed down, ultimately, down the electron transport chain. But in the process, we do get a little bit of ATP as well. So you can see that we have ATP being generated down the bottom. Here's the summary that I want you to know. So we have pyruvate in the cytosol. That's the result of glycolysis. One of the carbons is going to be pulled off, and that gives us a carbon dioxide. The acetyl group that remains is stuck to CoA. And in that process, in that transfer, we also manufacture NADH. Note that in this diagram, it's just showing one NADH and one carbon dioxide, but remember this is going to happen for 2 pyruvate molecules. So we get 2 NADH. The next step is that coenzyme is going to transfer that acetyl group into the citric acid cycle. The carbons are going to ultimately be converted into carbon dioxide, and we're going to get NADH and FADH produced, and also ATP. Again, just remember that we're doing the math for 2 starting pyruvate molecules. So all the numbers that I give you for glycolysis, for the citric acid cycle, and for the electron transport chain will refer back to 1 starting molecule of glucose. Just to remind you, we've got 2 membranes in the mitochondria. And we've got a space between them known as the intermembrane space or intermembranous space. And it's a bit exaggerated in this diagram. The inner membrane and outer membrane are quite different. The outer membrane resembles other membranes we see in eukaryotic cells. The inner membrane resembles bacterial membranes. And there's a reason for that. The mitochondria is thought to be a bacterium cell that became permanently incorporated into eukaryotic cells. And I won't get into the details of that, but there's lots of evidence to support this. For one thing, the mitochondrion has its own DNA. None of the other organelles have their own DNA. And also the little electric motors that we're going to talk about in a moment that use a hydrogen proton gradient to generate ATP are similar to little electric motors that we see in bacterial cells. So we have had electrons that have been pulled off of food molecules in glycolysis and stuck to NAD positive to make NADH, and stuck to NAD positive and FAD during the citric acid cycle to make NADH and FADH2. Those are our electron carriers. And those electron carriers are finally going to pay off. They're going to release electrons, and the electrons are going to pass down the electron transport chain. And that's what you're seeing here. So we have proteins that are embedded within the inner membrane of the mitochondria. So we've got NADH giving up electrons, and we can see that here at the beginning, and those electrons are going to pass through this electron transport chain, so from protein to protein, and eventually those electrons are going to be combined with 2 hydrogen protons and oxygen to make H2O. So if we take 2 hydrogen protons, add an electron to each of them, then we have hydrogen atoms, we add that to water, we get H2O. And the same thing is going to happen with the electrons that will be given up by FADH2. So the same thing, those electrons are going to pass along that electron transport chain. They're going to be combined with hydrogen protons and oxygen to make H2O. What happens as the electrons bounce from protein to protein is that they impart a little bit of energy that can be used to pump hydrogen protons from the matrix out to the intermembranous space. So we're going to have electrons that are going to engage in redox reactions. Energy is going to be liberated, and that energy is going to be used to pump hydrogen protons from the matrix out to that intermembranous space. And what's going to happen as a result is the intermembranous space is going to become full of hydrogen protons. So now we have a concentration gradient across the membrane. We have lots of hydrogen protons in the intermembranous space and far fewer in the matrix in the middle. Some of these hydrogen protons were hydrogen protons that were liberated during the process of the Krebs cycle. Remember, we talked about how hydrogen protons were a byproduct of some of those reactions. Now we have this strong gradient, and two things are going on. First of all, we have a chemical gradient, and as you know, things tend to move down their chemical gradient. But we also have a charge gradient. Hydrogen protons have the same charge. Like charges repel. They're going to try to push each other back into the matrix. But there's only one place where that can occur. There's only one place where hydrogen protons can flow back into the matrix. And that's through a complex set of proteins known as ATP synthase. There's a channel in the middle of this molecule, and the hydrogen protons can flow back through that channel. The energy that's stored as potential energy in that gradient is used to manufacture ATP. And this is chemoosmosis. Chemoosmosis, we're dealing with molecules, or in this case, protons that are moving down their concentration and charge gradient. In essence, the mitochondria becomes a battery. These hydrogen protons can't flow across the membrane readily because they will just stick to the hydrophilic heads of the phospholipids, but they can move through this channel. And as they do, they cause this complex structure to rotate. So we're converting this electrical energy into mechanical energy. And that mechanical energy is used to take ADP and inorganic phosphate, slam them together, and make ATP. ATP synthase is, in fact, an electric motor. It's a tiny electric motor. In this diagram here, the different colors are showing us different proteins. So this is a complex structure. And a lot of researchers that are interested in nanotechnology, for instance, and making nanobots and things like that are intensely interested in ATP synthase because they would like to replicate this structure. This is an animation of how ATP synthase works. We've got the inner membrane shown here in gray. Above that is the intermembrane space, and below that is the matrix. Up in the intermembrane space, you can see little yellow cubes which represent hydrogen protons. What's going to happen is hydrogen protons are going to flow down their concentration gradient and down their charge gradient through a channel in ATP synthase. As they come into ATP synthase, they'll slot into little grooves in this rotor. That's the rotating piece. So as a new hydrogen proton comes in, it's going to repel the one that's already there, and it's going to cause the rotor to turn. The protons will go around one full circle, and then they'll leave through an exit channel. So you can see the exit channel there where the protons are moving back into the matrix. The protons can be reused. They can be pumped back out into the intermembrane space by the electron transport chain. The rotor rotates, and as it does that, it rotates a shaft. And that shaft is going to change the shape of these proteins, these lobes, that make up part of what's called the lollipop head. These proteins are going to grab onto inorganic phosphate and ADP, and as they move, they will take those two components and slam them together and make ATP. So we're taking an electrochemical gradient, we're converting that potential energy into mechanical energy, energy of rotation, and then we're using that to slam two things together to create potential chemical energy. And what you're seeing in this figure here is the complexity of these proteins. So you're seeing the structure of each of the proteins involved. It really is quite an amazing complex of proteins. The steps that occur within the mitochondria require oxygen. So in the citric acid cycle, we're pulling carbons off of the acetyl group that enters into the cycle, and we're taking those carbons and sticking them to oxygen to make carbon dioxide. So of course, we need oxygen. With the electron transport chain, the terminal acceptor for electrons is oxygen. So what I mean by that is electrons are going to be released by electron carriers, and they're going to be transferred from one molecule to another in the inner membrane. But eventually we have to do something with those electrons. We have to get rid of them. And what happens is they're stuck to oxygen along with two hydrogen protons to make water. And we say that the oxygen is the final electron receptor, because of course those electrons are going to be pulled closer to the oxygen in the water molecules that are created. Incidentally, about 10% of the water that you use in your body, you made yourself. You created it by this process in the cells of your body. Okay, but what if there is no oxygen present? So we don't have oxygen. Well, we can't do the electron transport chain because those electrons are going to build up. They'll have nowhere to go. So we're going to get this backlog of electrons in the electron transport chain, and it will shut down. And again, if we don't have oxygen, we can't pull carbons off of the intermediates within the citric acid cycle to make carbon dioxide. If there's no oxygen available, the electron transport chain will no longer accept electrons from electron carriers. There's simply no place for those electrons to go. And you might think, well, glycolysis can still continue, but actually it can't. And the reason for that is there is a step in glycolysis, if you remember, that generates NADH. So NAD positive is going to accept electrons and protons to become NADH. And that step has to occur. If it doesn't, glycolysis won't move past that point. Glycolysis will shut down. So without removing those electrons from NADH to regenerate NAD positive, your cell will eventually run out of NAD positive and glycolysis will quit. So those electrons have to go somewhere. They have to be pulled off to regenerate NAD+. And what a lot of cells can do in the absence of oxygen is they can take those electrons and stick them to something else. Pyruvate is a good target because pyruvate is not going to be used by the mitochondria. So in yeast cells, for instance, if they have oxygen, they can undergo all the steps of cellular respiration. But if they don't have oxygen, they'll take the electrons off of NADH, stick them to pyruvate, pull off a carbon, stick that to oxygen to make carbon dioxide, rearrange this to make acetaldehyde, stick the electrons to that to make ethanol. So yeast manufacture ethanol. They also give off carbon dioxide in the process. This process is known as alcohol fermentation. We can't do this particular type of fermentation, but yeast cells can, and they generate the ethanol that we drink in beer and wine, etc. But remember, the whole point of fermentation is to pull electrons off of NADH and regenerate NAD positive so that at least glycolysis can continue. We can do something similar. There is a type of fermentation that happens in some human cells, and that's lactic acid fermentation. So in muscle cells, for instance, if they're deprived of oxygen, they can, for a very short period of time, undergo fermentation, where they will stick the electrons to pyruvate to regenerate NAD positive and in the process generate lactate. So in yeast, fermentation produces carbon dioxide and ethanol. The carbon dioxide produces bubbles that cause bread to rise. It also produces the bubbles in beer and champagne. The ethanol is what gets you drunk when you drink vodka or beer or wine or whatever. Incidentally, bread is not going to get you drunk because the ethanol is going to evaporate. In humans, we don't produce ethanol, otherwise we would be drunk quite often. Instead, we produce lactate, which is converted into lactic acid. But again, we can't do that in all of our cells, and we can't do it for very long. And the reason for that is that the energy output from fermentation is much, much lower than for aerobic respiration that occurs in the presence of oxygen. For glycolysis, we only get 2 molecules of ATP produced for every glucose molecule that we start with. And for aerobic respiration, we get 38 molecules of ATP. This PowerPoint does contain a bit more information than I expect you to know, but I do want you to know the inputs and outputs of each of the 3 processes that make up cellular respiration. So for instance, with glycolysis, what goes in, what comes out? How many electron carriers are generated for each molecule of glucose that comes in? For the Kreb cycle, again, what goes in, what comes out? How many molecules are produced for each starting glucose molecule? Remember that metabolism consists of 2 categories of reactions. We have catabolism, where things are broken down and we generate energy in that process. That's because we take complex molecules, break them apart, break bonds that tend to be nonpolar, and then we reform new bonds that are polar. We get more energy back when we create those simpler molecules like carbon dioxide and water. Anabolism is where we synthesize complex molecules, and that requires energy. All reactions in a cell, with very few exceptions, require enzymes to catalyze those reactions. And remember when we talked about enzymes last term, what they do is they bind to specific substrates, they bring them together, they lower the activation energy required for those reactions to occur. Enzymes can be inhibited. We've talked about that before last term. We'll come back to this again later in the course. But ATP, remember, is the energy currency of the cell. Ultimately, we're going to use potential energy to build ATP. We're going to take ATP apart to release energy for anabolic reactions. I've tried to reduce the basics to this slide here. This is by no means everything you need to know, but it's a good starting point. Again, another diagram that kind of reduces it to the very basics. Not everything you need to know is here, but a good starting point to understand what's going on in these different processes. At the very least, from this lecture, you've learned why you need to breathe in oxygen and why you breathe out carbon dioxide. A few study questions, I'll let you think on these. And finally, I want to stress again the importance of this diagram. It's a really nice summary of what's going on. We're going to be talking about muscle cells in our upcoming lectures, and it's important that you understand this in order to understand how muscle cells work. because there are some situations where muscle cells are starved of oxygen and they undergo lactic acid fermentation. There are different types of muscle cells that utilize cellular respiration in different ways. Now there's one other thing on this diagram here that I didn't get into, and that's this bit here. So notice that this transfer of the acetyl group off of pyruvate into the mitochondria can liberate energy. In some cases, it can be used to build ATP, but that depends on the molecule that's used to shuttle that acetyl-CoA into the mitochondria. We're not going to get into that level of detail, so we can just ignore that. Let's just assume that there isn't any ATP produced there, and that means that during aerobic respiration, cellular respiration in the presence of oxygen, we have a total of 38 ATP being produced. 2 from glycolysis, 2 from the citric acid or Kreb cycle, and 34 from the electron transport chain. Okay, and I'll leave it there. Again, this is important stuff that I'm hoping you have some familiarity with. If you don't, review, because this will be important coming up when we talk about muscles.